Can you hear me now?
Yes, sir.
Okay, good. Yeah, so yeah, I'm really sorry. So we had midterm interviews this week, so it has been, and that was something I didn't plan for. But anyway, so just go start with this. Yeah, so as I said, that it's kind of hectic, but we'll start the vision system today, and the auditory and somatosensory system will go over on Monday. So we'll be done with that, and then the next two segments would be motor systems and emotions and cognition and emotions, which we'll try to cover in last 3 weeks, and then what we'll do is try to do last one or two classes. We'll go over some of the experiments that we do. Either I'm not an expert in experiments that are done on monkeys or humans, but I can go over them. That what we do in a laboratory, like say at the Center for Neurosciences in IISc, and also the experiments we do with mice and in culture. So we'll just go over the basic techniques, not basic techniques, but the kind of the possibilities that are there right now in neuroscience, so that you kind of find it interesting to study. Yeah, and so today we'll just so also make it pretty quick. Yeah, so we'll go to the vision system, and of course, it involves the eye. And then what you have here when you were looking at somebody's eye, you have the pupil at the center. That's the what the light is going through, and then it's surrounded by the iris, which is the gives the color of the eye. And the iris has the muscles that would constrict or dilate the pupil, and that basically is deciding how much light to allow. And also we'll talk about it later that there are other systems like these are connected to the brain. Yeah.
I'm sorry, I was trying to speak earlier, but my network was not working properly. That's why I wasn't able to. You were saying, so how many classes are left for this module?
Oh, so this is the sixth one, so totally 10. So there are, so 1 second. So there are 4 more basically.
So in the next week, we'll be having 4 lectures. Is that it?
Yeah, 3. Right. So Monday, no, totally 4. Yeah. So Monday, Tuesday, Wednesday, Thursday. Yeah, 4.
Okay, thank you.
Yeah. So we missed. So there were 3. So one basically is missing, right? And then if you guys have questions, then we can do some other Saturday or something. Does it sound good?
Also, sir, you had said that you'll be just briefing us about the mode of assessment. So I'd forgotten to ask you about this previously. So have you finalized anything about that?
I actually haven't put. This is my first time. I don't even know. Like I have to see what other people are doing before, or I can talk to Jean and the other the teaching assistant, and we can whatever was last year. We can just go with that. I haven't looked into it. We can talk. I can talk to the TAs, and then we can let you guys know by tomorrow or something.
Okay, so thank you.
Yeah. Are there any questions? Yeah. So the people, the light goes through that, and then we'll talk about later that how the system is connected to the brain and how basically the light is going through this, hitting the retina, and it's transferring to the brain, and there is another circuit which coming back and telling it's constricting or dilating. And the cornea is the glassy layer that's basically covering the entire structure. And the sclera is the white part that is continuous and it's giving the shape and it's making like a kind of the hard structure that the eye is. And there are muscles behind this, and there's a conjunctiva, that the red layer. And because of the muscles, because of the conjunctiva, you really can't see the muscles, and that's kind of moving the eye left and right. and the muscles here are constricting the pupils and or dilating the pupils, dilating the pupil. So if you kind of look, kind of take a cross section and look at the eye, it would look something like this, where again, what you said is there is a cornea, that is the topmost layer, and then there is something called aqueous humor, this liquid. So the cornea itself doesn't have any blood vessels or vascularization, so it's getting all this nutrition. So this is a living structure. There's not a dead structure. So it needs nutrition, and the nutrition it gets is from the aqueous humor that is just beneath the just beneath the cornea in front of the pupil. And then what you have is the iris that is that's kind of controlling the diameter, and the light is entering through the center. And behind that you have the lens that is basically kind of focusing the light onto the cornea. And there are fibers which are called the zonule fibers or ligaments that are attached to the ciliary muscles here. So these are the ligaments and the ciliary muscle, and that's contracting, and it can change the diameter of the lens, and depending on where you're focusing your vision, it can this diameter will change, and then say like you are close by, it will be much thicker, you are far away, it will be more slender, and that will be controlled by the ciliary muscles, and the light will accordingly get focused on the cornea. So on the retina. And retina is basically there's in the center there's a liquid-filled structure which is called the vitreous humor, which is kind of a jelly-like structure, and these are all transparent, so the light can go straight through, and they're optically transparent. And then it hits the retina, which is basically the structure which contains all the apparatus that you need to transform the optical the light that is coming into electrochemical energy or electrochemical signal that eventually this retina will send to the brain, and the brain can translate what is happening, the information it's getting, into the image that you perceive. So the entire kind of the neuroscience is basically here. Everything else is in front, but this is all controlled, and the light is coming and getting focused here. And the fovea is right in the center, which is known as is a small structure that forms the clearest image, which will come to it a bit later. Yeah, so then the image formation. So how the image is formed. So basically the light is refracted through the lens and through the vitreous humor, and is focused on the fovea. And the focal distance, which is so basically the focal distance is the distance between this, and that is around like 2.4 centimeter. The distance starting from the start of the cornea to the point it needs to be focused. And the light, the more it goes far away, it gets basically refracted this way, and it's focused here. And the refractive power of the eye is generally defined as something called diopters, which is the inverse of the focal length. So focal length, generally, usually the eye, on an average, the eye focal distance, not focal length, focal distance is 2.4 centimeters. So the diopters is 42. The refractive power of the cornea is equal to 42. That's how it's defined. And there, as I was talking about, there is something called pupillary light reflex. So the pupillary light reflex is basically, say you are standing in a dark room and suddenly somebody turns on the light, and your pupils is actually it will constrict really fast, and which is known as the pupillary light reflex. And this is entirely consensual, which means that even if you're focusing the light on one eye, the other eye the pupil will constrict, which means that they are somehow working together. So they are actually in the brain. If you see their circuits, so the information is coming. The afferent neurons are from the retina. They are coming into the brain in this region called the pretectal area, and eventually there are motor nerves here information will get processed and go out, and then they are connected to the pupillary sphincter or the muscles that are controlling the diameter. So what happens is that even though you're focusing light on one eye, the other pupil also getting constricted because the processing is happening inside the brainstem in the connections between the predictal area and the edingovascular nucleus. You don't have to know the nuclear what it does, but the idea is that it's important to know because if it's not happening in the sense that pupillary reflex or the pupil diameter between two eyes are not in sync with each other, that means that you have a neurological disorder which is affecting the brainstem. Often can be, sometimes can be a stroke or injury to the brainstem that can make the pupil diameter not to be same with each other. And the nuclei that controls this is called the olivary pretectal nucleus. It's very interesting because if you record from these neurons, so the firing rate of these neurons is directly proportional to the intensity of light. So you can do this experiment in mice or rats, and what they do is they make a preparation where you have these optic fibers or the retina intact, and you have the brainstem, and they are and this is not a living animal, so they have taken the brain out, the head out, and then they have dissected out, and it's all bathed in a solution that will kind of make the cell survive for some time. And then they're recording from that, and they see that as you change the intensity of the light, these neurons are the neurons are firing more and more as it was stimulating it by light. Yeah. So, and then of course the biggest question is, how is the light transformed into a signal that is perceived by the brain, and what happens at the retina, which is kind of receiving all the information all the light information from outside. So at the very so it's also kind of interesting that it's arranged inside out. So what you would expect is that the photoreceptor should be the outsidemost cell type, and the neurons that are transmitting the information to the brain should be the insidemost. But it's the opposite, and because all these cells are transparent, it doesn't really matter that much because. But so one of the ideas that why is that is because of it protects the photoreceptors, and there is a layer beneath the photoreceptors that is important for maintaining this. Yeah. Do you have a question? Okay, so yeah, so it's maintaining the photoreceptors, and in cats and dogs, this layer is basically reflective, and that's why their eyes glow in the dark. And so the photoreceptor, the most insidemost structure, but this contains all the photopigments that are kind of transforming the light into a potential change in potential that is transmitted through the other cells. Yeah, and then what you have is a bipolar cell that is connecting the photoreceptors and the ganglion cells, and it's very important to remember that these cells can depolarize and hyperpolarize. So, which means just like neurons, the potential will drop and rise as the signal comes in, but none of this fire and action potential. So the action potentials are only fired by ganglion cells, and ganglion cells can kind of transmit this information from the bipolar cells to the brain. And then you also have neurons called, or non-neurons, cells called horizontal and amacrine cells. So these are cells that are basically, amacrine are connecting the photoreceptors and bipolar cells with each other. So they are mediating the information. So these cells can influence what you see. So they can also make each other make each of the cells to talk to each other. So they can decide, okay, like this signal is the strongest, so this is something. That's how we choose between, even though there are two things in our visual range, but we can give importance to one and versus kind of ignore each other at will. Or it depends on the color or the distance it is in. So what we're focusing on. So everything, those kind of events or the physiological events are done, are computed by something, cells like horizontal cells, amacrine cells. So amacrine cells are connecting between the ganglion cells and bipolar cells to each other. And these are somewhat like, the interneurons that you would see in the brain or in the spinal cord, we have spoken about before. Yeah, so and the anatomy, so you have basically the rod and cone photoreceptors, which we are here, and then you have the ganglion cells, and which are the only outputs. And as I said, the ganglion cells are the only ones that can fire action potential and take the information to the brain. Yeah, and so as I said, the structure is kind of opposite to what you would expect. So the photoreceptors are the innermost, and you don't need to know all the layers here, but the idea is that it's a laminar structure, which means there are layers of cells in the retina, and then when you look at the foveates come later, like the structure a little bit changes. Otherwise, it's pretty much uniform all over the place. So that's the only place that kind of transmitting the information. And then you have the rods and cones. So rods and cones are basically the only elements that are actually classically that are actually sensitive to light. So the rods and cones are connecting to bipolar cells, and if you look at the structures, that you have the synaptic terminals that are connected to the bipolar cells, you have the cell bodies of these neurons, and then you have the outermost, the cone, which is called the membranous disc or the disc-like structures. So the cones have lesser number of discs, the rods have higher number of discs, and the more the number of discs are there, the more amount of photopigment. So when there is a bright light, you basically have, you can use your the cone photoreceptor, but when you have a lower light, then you can use your rod photoreceptor. So a combination of both actually kind of allows us to adjust to the amount of light that is available. And so this is a electron micrography, which is in the textbook, and they show that how the rods and cones kind of look quite different from each other. So rods, you have this thin elongated structure, cones are much smaller, something like that. And depending on the structure, that's how they're named. The electron microscopy structure, that's how they are named. And this is called something known as the scanning electron microscopy. So in the scanning electron microscopy, what you basically would see that the outside structure. So if something is called a transmission electron microscopy, you will see it is a much higher resolution, and just like the synaptic vesicle pictures I had shown you before, so that you will see the actual cell organelles and the minute structures inside the cell. But scanning is basically, it's not penetrating, so it will show you a three-dimensional structures. Yeah, and then as I told you, that the fovea is where all the lights are focused, and then you have the outermost ganglion layer, the inner nuclear layer, and the outer, just like the laminar structure we're spoken about, and then towards the inside that you have the rods and cones. And this ganglion, the bipolar cells and the ganglion cells, they're kind of move apart to expose the cones, and so the light intensity that the cones get is the maximum here, and that kind of it's known as the foveal vision. So in the foveal vision, it's kind of centered. So if you're focusing on something and the center of the image form, it's basically perceived by the rods and cones in the fovea region of the retina. And so what are the experiments can be done? I mean, there are quite a few, but one of the interesting experiments that you can do is kind of find what is the receptive fields, what is the receptive field of the particular eye. And what people do is, you can do this in monkeys or rats, where you record from the optic nerve, and then you shine like very precise light on different parts of the retina or different parts of the eye, and then you can see if the neurons are firing. So what you do is you record from the ganglion cells of the optic nerve, and to see what is the receptive field, you go on shining the light at different areas, and you kind of map, that one neuron, or like from kind of one or two neurons from the nerve that you're recording from, where exactly they have their retinal cells. So, and this idea of mapping the receptive field is not limited to the visual system. Same thing happens in somatosensory system, or in touch and pain, where you basically want to find what are the nerves that are innervating a particular patch of skin, and then you kind of either you can kind of take a pin and prick like a small area, you go on like in a pattern, in a grid pattern, and then see the neurons that are activated. And once you are in the kind of receptive field of that neuron, you will know that okay, this neuron is one or two neurons in a nerve is basically innervating this part of the finger in the case of somatosensory system, or this part of the retina in the, or one nerve is basically one kind of ganglion cell is innervating that part of the retina. Yeah, and then so as I said before that most neurons, most ganglion cells these are the neurons that are carrying information from the from the retina to the brain, and the neurons themselves are not photoreceptive, and they have to get information from the rods and cones. And there are these cells called IPRGs, or intrinsically photosensitive retinal ganglion cells. And these are something that we discovered pretty recently, and you can go and read a bit about it. I can send you guys papers about it. It's kind of fascinating, because the rods and cones have always been the thought about the only photoreceptors, and this is something that is discovered, say, around 8 or 10 years back, or something like that, or it might be 11, but I can send you as a paper, as I said. And so the scientists have found that there are ganglion cells that can process light directly, and that's why they're called intrinsically photosensitive. And they don't need the rods and cones or the bipolar cells, and they are tuned to a broad range of light. And that's how, like, even in the dark, you can have some kind of light. Even very low light, you can have some kind of photosensitivity, or you can even get some kind of, your brain can get some kind of information, even when it's kind of below the range what rods and cones can detect. And so that's that. And so then you have basically the bipolars and the IPRGCs. So, and then something I would like to show. Yeah, so yeah, so that's basically the vision system, and then we'll do the what do you call it? So then we'll do the motor system. No, rest of the sensory system on Monday, and we'll do the motor system. And so one day emotions, cognition, and the last day would be So and the last two can be questions and show you some data and some recordings and some kind of newer imaging tools that what people use. Yeah, so that would be that. And then any questions? Yeah. Any questions? Yes, Tanish.
I'm not sure, but I read somewhere that in octopuses, the arrangement of the photoreceptors and the bipolar cells and ganglion cells, that is reversed.
Okay, I didn't know that.
Uh, so why would that be?
In octopuses, sorry, can you repeat the question?
Like you said that in human beings and in mammals, that the photoreceptors are the innermost layer and they are not directly exposed.
Right.
So I read somewhere that in octopuses it's the opposite case.
Right. Yeah, I'm just Googling it. I didn't know that. Yeah, so it is opposite. What is it?
So you told us that
So they don't have what do you call a physiological blind spot for vertebrates. So in the cephalopods, basically all the cephalopods, including octopuses, have opposite. So the nerve it doesn't pass. So our thing is that you're right. So it's opposite, right? So the nerve actually passes over the retinal cells, all over the rods and cones, over the photoreceptors. So there are parts of uh, our eye, our vision system has basically blind spot that are like we have a field of view and then we can see after that. Even within a field of view, we have a corner blind spot because of the nerve passing on it, but they don't have the opposite. So it's like a camera, basically. So yeah.
yeah. So my question is that like you told that in us the photoreceptors are the innermost because they are like protected from the outside environment. I mean
Yeah, I guess the range of vision is more, and that's their requirement of finding food or just be careful, like kind of watching so that they don't get eaten up, right? And they have this huge eyes, so I mean their vision system is kind of what to say, in a way pretty developed, highly developed, so that I guess protecting the eye is not their primary concern, and giving them wide range of vision is more important.
Okay, thank you.
Yeah, I don't think even they have like an answer for that. I mean, there are, I guess, arguments why they have this camera type eye that. So in cameras, they don't have because they don't have nerves, right? So you can, what is the chip size? That's your kind of visual range kind of thing. But yeah, so I think it's like debated. I don't know. I don't have the answer for that. Why that's the case? If you ask me. Yeah. Anybody else? Okay, so should we just stop it there and then get back on Monday? So Monday, like I might take 10 minutes more than an hour, so if that's okay. Okay. Okay then. All right.
Sir, I had a question with regard to the previous class.
Oh yeah. So yeah, I can email. So you email me. I just didn't get the time. I'll email back. Is that the
Okay,
question?
thank you. Yeah.
Yeah. Yeah. I'll do it today. So all right. All right. Thank you. Bye.
Thank you.
Yeah.